王老师，我们这个的话，在 YouTube 的话也是有直播的，然后我们一会儿的话，您就直接开始，然后我先介绍一下您，然后的话就是我们最后是有问题的话留在留言区，然后我们一起回答，好吗？
好的，我可能就
嗯，好，
是说讲的时候可能用英文，然后讨论用中文，因为我感觉
，OK，可以
about science is very difficult，就是 struggle between
，OK，
English Chinese translation. So, sorry。
好的，好的。
常总，你再把这个会议再到各个群里再转发一下，再让其他人帮忙转发一下。
嗯，我跟大家再 share 一下。好了，我看时间也差不多了哈，那我们就先开始吧。然后后面的话，大家也会陆续再进来。我们今天的话呢，就我先给大家介绍一下，王昭明老师。那王昭明呢是圣约达儿童研究医院的副研究员，主要任职于流行病学与癌症控制系，同时也在计算生物学系。那王老师呢，主要是在嗯。这个癌症学会、癌症检测和进展评审委员会当中担任常任成员，然后呢也是 NCI 资助的研究员，拥有3个正在进行的201项目，从2007年以来发表了220多篇的论文，并引用量呢也超过了38000次，谷歌学术 H 指数为85。那我们接下来的话呢，把时间交给王老师，然后大家有任何问题的话，可以在留言区，然后呢，我们最后一起提问。另外呢，王老师这边也在招聘博士后，然后有兴趣的话呢，也欢迎和王老师直接联系。王老师，那您直接开始好吗？
好的，谢谢常龙。Um, I will use English for the presentation, and then we can use Chinese to for the Q&A. So one third of my research portfolio focuses on the topic, which is aging biomarkers and accelerated aging in survivors of childhood cancer. I will start with a couple of slides to orient you to childhood cancer survivorship research. Due to advancement of cancer therapy, greater than 85% children diagnosed with cancer will survive 5 or more years. About 13,000 out of 15,000 newly diagnosed cases will become survivors each year. The total number of survivors in the US is now over 500,000, which translates to 1 in about 750 is a childhood cancer survivor. Survivors are at risk for a broad spectrum of adverse outcomes due to cancer and treatment. Although there are some well-established links between the treatment and the led effects, not all risks can be accounted for by treatment exposures, and that's where genetics and epigenetic factors may come into play. My research primarily uses resources from the St. Jude Lifetime cohort. It is a hospital-based retrospective cohort with prospective follow-up, included 5 or more year survivors, included patients diagnosed between 1962 to 2012 and younger than 25 years, included all diagnoses. Cancer treatment exposures were extracted from the medical records. And we use clinical visits and surveys for the methods of contact and use clinical assessment, medical records, self-report, and National Death Index for ascertainment of outcomes. Blood is used as the source of germline DNA. So we know aging are multifaceted, including different biological mechanisms. For example, these 4 primary hallmarks are deemed as causes of damage, including genome instability, telomere attrition, epigenetic alterations, and loss of proteostasis. And the hallmarks of aging have been recently expanded to include these 3 additional: disabled macroautophage, chronic inflammation, and dysbiosis. My colleague, Dr. Q-Ness, really pioneered aging research in survivorship about a decade ago, reported frailty as a sign of aging acceleration among survivors. The conclusion from this study is that the prevalence of frailty among young adult cancer survivors is similar to that among adults 65 years old, which translates to accelerated aging of approximately 30 years. Pre-frailty and frailty were defined as 2 and 3 of the following 5 conditions, which include low muscle mass, self-reported exhaustion, low energy expenditure, slow walking speed, and weakness. As you can see here, for the CNS tumor survivors, about 40% have either pre-frailty and frailty. The extent of the pre-frail and frail condition vary across different cancer diagnoses. This really highlight the accelerated physiological aging among these survivors. My group, over the years, published this series of studies. The first is about leukocyte telomere length, and the second is about epigenetic age acceleration, and the third is a genetic association study using epigenetic age acceleration as a molecular trait. And a couple of latest publications, including epigenetic age and epigenetic age acceleration among children and adolescent survivors, as well as deficit accumulation index and aging biomarkers. Earlier this year, we published association between health-related quality of life and aging biomarkers, again in the JNCI journal. Dr. Nansong, my first trainee as a postdoc, published this article reporting shortened leukocyte telomere length associates with an increased prevalence of chronic health condition using St. Jude lifetime cohort. Dr. Kyle Walsh, an expert in the telomere research, wrote a very nice commentary highlighting our study published in the same issue of the Clinical Cancer Research. In this study, we found leukocyte telomere length in survivors was significantly shorter than controls overall and across diagnoses. When projecting onto the linear regression line of the leukocyte telomere length against age, the leukocyte telomere length of 6 kilobases corresponds to age of 37 years among survivors and an age of 48 years among controls, suggesting an acceleration of aging by 11 years. We carried out a coronal hematopoiesis study last year, including survivors and controls from St. Jude Life cohort, using ultra-deep sequencing of the coding region for 39 coronal hematopoiesis candidate driver genes. We found the prevalence of CH mutations detectable at variant allele fraction of 0.1% was significantly higher in survivors compared to the controls. And the overall prevalence was 15% versus 8% across the different age beings. So the elevated CH rate of the survivors generally corresponds to that in the controls about 20 years later. So remember, you have 30, now 20, and previously epigenetic measurement, you have about 11. So really depending on what kind of metric you look at, you will have sort of the different accelerated aging between the survivors and the controls. Telomere length has been well established and well studied as an aging biomarker. However, in this review paper, the authors show that telomere length has a much lower predictive power than epigenetic clock. So with this in mind, let's switch gear to another aging biomarker, epigenetic age acceleration. This research project was led by my former predoctoral trainee. There are at least a dozen epigenetic clocks have already been proposed. Here listed are 4 popular ones. Hannam and Horvath are the first generation of the clocks selecting CBGs to predict chronological age with smaller number of predictor CBGs. Levin and Grimage were among the second generation, attempting to model physiological measurements and health outcomes with larger number of CBGs. In this study, we adopted Levin's clock, PhenoAge, to calculate epigenetic age in this published study. The concept of epigenetic age acceleration can be illustrated with this cartoon. The solid blue line shows how epigenetic age changes with chronological age. The rate is very fast in the first year of life. Afterwards, it decreases gradually in a nonlinear manner until about 20 years of age, and then gets to a slower constant rate. Individuals above the line had positive epigenetic age acceleration and are older than their peers at the same chronological age. Conversely, individuals below the line had a negative epigenetic age acceleration and are younger than their peers at the same chronological age. The correlation between the epigenetic age and chronological age is high, 0.88 for Pearson correlation for both survivors and controls. However, the age slope, that is the annual change of methylation age, is 1.24 in survivors and 1.08 in controls, which is statistically significantly different. We found epigenetic age acceleration is much higher in survivors, shown in red, and the controls shown in blue. Survivors of Hodgkin lymphoma seems to have the highest epigenetic age acceleration across all primary diagnoses. Survivors with chest radiation, abdominal pelvic radiation, or exposure to 3 different chemo agents had significantly elevated epigenetic age acceleration than non-exposed survivors. We compared survivors with 5 different modifiable health behavior, including tobacco use, smoking, diet, physical activity, alcohol use, and resistance training. Due to the multifactorial nature here, we defined 3 groups, including 4 or 5 good behavior, and we grouped it as favorable, 2 or 3 good, intermediate, and 0 or 1 good, unfavorable, based on these 5 components, and observed the epigenetic age acceleration trended upward among survivors with favorable, intermediate, and unfavorable health behaviors. 6 chronic health conditions were significantly associated with epigenetic age acceleration after adjusting for other factors. There are hypertension, myocardial infarction, obesity, peripheral sensory neuropathy, obstructive pulmonary deficit, and obstructive sleep apnea. So let's look at the 2 age acceleration model, and we can see that the aging trajectory for survivors varies depending on the dynamics of molecular marker. First, telomere length show a pattern of accentuated aging with the initial hit, but a parallel age slope. Whereas epigenetic age acceleration showed a pattern of accelerated aging with a faster pace or steeper age slope. To conclude, for this project, epigenetic age acceleration is higher among survivors than controls. Epigenetic age acceleration is elevated among survivors exposed to treatment or unfavorable health behaviors. And EAA is positively associated with increased rates of certain chronic health conditions. And clinically, EAA can potentially be used as a biomarker and targeted for future intervention studies. Let's move on to the next published study. And this research project was led by my former postdoctoral trainee. With the expanded DNA methylation profiling, we conducted a genome-wide association study and discovered a strong HLA locus associated with epigenetic age acceleration based on Hannum clock. It was replicated in the second set of survivors, but in controls, but not in controls, suggesting these genetic finding may be survivor specific. We also found an association at SELP gene locus with epigenetic age acceleration based on Horvath's clock. As you may know, SELP gene encoding P-selectin is a well-known hematopoietic stem cell marker and functionally involved in age-associated HSC functional decline. This signal was replicated in survivors and also in controls. Here we proposed a potential molecular mechanism underlying the association. On one hand, the risk allele C is negatively associated with the DNA methylation level. So itself is a methylation QTL. And positively associated with the gene expression level of SELP. On the other hand, survivors exposed to the RT radiation and have lower methylation level of these CpG and higher expression of this gene. So this gene is a ligand for the SELP, so the SELP ligand. High expression of both genes, so the elevation of these and these ligand, both are pro-inflammatory, and survivors who receive both RT and carry homozygote or heterozygote risk allele for SELP are the survivors with higher epigenetic age acceleration. So from this cartoon, you can see that the genetic and environmental effect on the accelerated aging for the survivors. And let's move on to the next project led by my other postdoc trainee. The motivation for doing this is that the previous study demonstrated the cancer therapy are strong risk factors for the aging acceleration among survivors, and that EAA is associated with chronic health conditions. But this data, the previously published data, included few survivors with age measured younger than 20 years, so limiting the ability to evaluate early epigenetic aging after the cancer treatment. So for this study, we included younger people, younger than 20 years old. And now we have this part, and this part was missing in the previous published study. And you can see that the change of epigenetic age is much faster in children and adolescent age groups based on the Levin clock. And when we observe, when we see the epigenetic age acceleration, there's subtle difference among these three age groups, but the most striking pattern was lower EAA in both children and old adults compared to other chronological age groups. And we found EAA was significantly associated with risk of early onset, obesity, and severity burden of all the chronic health condition, and also the, more importantly, the late mortality. So this study demonstrated the annual change of epigenetic age, and EAA differ across chronological age, and EAA measured in children, adolescent survivors associated with early onset obesity, and all the CHCs and late mortality. So our finding really suggests potential early entry point So all these observational study provide a future intervention study introducing anti-aging intervention studies such as lifestyle modification and pharmacological intervention, including potentially the DNA methylation, demethylation agents that may reduce morbidity and mortality during the long-term survivorship care. And Ellen Williams, another former postdoc working with Dr. Kevin Krull, is now assistant professor and a collaborator, and she's now a tenure-track assistant professor at the University of Rochester. She led this recent publication. For those who is not familiar with deficit accumulation index, this is a definition. The DAI is a well-established clinical tool, including 44 aging-related items, such as chronic health conditions, functional, psychosocial, mental well-being. Items responses were summed and divided by the total number. So it's a fraction between 0 and 1. So the DAI results were characterized for easy interpretation of our results. And if DI is lower than 0.2, we categorize as low. And then the medium for DI between 0.2 and 0.35, and high for DI score greater than 0.35. And we found a very nice, just focusing on the right-hand side. So we actually analyzed both leukocyte telomere length and the EAA. You can see that the association, the dose response, very nice. So you can see that the adjusted mean of the epigenetic age acceleration, each of these, and very nicely correlated with the low, medium, and high DAI. So the DAI was significantly associated, but not the leukocyte telomere length. And you can see that probably sometimes the consideration of other biomarkers when you do aging study is also important. And both DAI and epigenetic age acceleration were effective identify aging phenotype, and either may be used or with telomere length to measure aging in response to intervention targeting aging pathway. We aim to expand into survivors of other race, ethnicity, and carry out trajectory analysis. And in the near future, using serial sample, this is one of my funded where I will collect it. Using the collected serial sample to look at the trajectory of the epigenetic age acceleration. And early this year, this paper was published. We looked at overall and domain-specific health-related quality of life. So a lot of epidemiology here. And we look at the correlation association with DNA methylation measures of biological aging. And so in this study, I just want to just quickly show that there is a strong association between, on the left-hand side, the different metrics of the health-related quality of life. And on the right-hand side are the aging biomarkers. So the links are the pairwise strong, significant, statistically significant correlations. And the key take-home message here is overall and domain-specific measures of health-related quality of life are associated with the DNA methylation measures of biological aging. And again, longitudinal study is super important, and we'll come back to this later. And this is a study that it's in press, actually. It's incidentally, today I'm doing the proofreading for this paper. Will go online very soon, and JAMA Network Open. So this is about racial disparity and rules of social determinants of health in epigenetic age acceleration among survivors of childhood cancer. And just, the definition of social determinants of health. Here, we only included very limited factors, personal level socioeconomic status, educational attainment, and personal income, and neighborhood-based socioeconomic status variables can be measured by area deprivation index. and ADI was treated not only as continuous, but also as category variables. And importantly, the BMI and smoking status were adjusted as covariates. So I'm going to skip the statistical analysis part, but I think that just really briefly mention the mediation analysis. It's a biostatistical approach to sort of analyze the mediator between the causal, between the exposure and the outcome. So here we model the epigenetic age acceleration as the outcome, and then the exposure was the race ethnicity as the exposure, and the SDOH as a mediator. So overall, when we compare epigenetic age acceleration between the non-Hispanic Black and the non-Hispanic white survivors, we found that epigenetic age acceleration is significantly greater in Black, shown in blue bar, versus non-Hispanic white, shown in this red bar, with the adjusted least square mean being about 1.41 year and 0.46 years, respectively. So in the overall analysis, I just look at the red box here. So it's 0.94. It's just 1 year in difference on average. So when we stratify by the race ethnicity, they look at the stratified model, the non-Hispanic white and non-Hispanic Black. We see that the different treatments have different impact. For radiation, the impact on the white is larger, 3.65 years versus 2.36 years, which is higher in white than in Black. But interestingly, we see the opposite for epipital phyllotoxin exposure. This is a chemo drug, and the increase of the epigenetic age acceleration is greater in Black, 2.39 years versus 0.68 years in the whites. So this table summarizes the mediation analysis. We found that the average causal mediation effect for of the race, ethnicity, epigenetic age association were mediated by educational attainment. It's a 22.7%. And similarly, for the ADI, the area of living, the neighborhood, is 22%. So our findings suggest that impact of cancer treatment exposure on EAA differs by race. Specifically, we found the association chest RT and EAA was greater among whites, whereas the association between epipital phyllotoxin and EAA was much greater among Blacks. And the racial differences are partially mediated by the social determinants of health, specifically educational attainment and area deprivation index, suggesting that improving social support system should be tested as an intervention strategy to mitigate socioeconomic disadvantages contributing to accelerated aging among childhood cancer survivors, especially Black survivors. I'm going to skip this. I think really quickly just mention that all the previous work we've done so far, we used the PBMC, which is a mixture of all these subtypes. And in a manuscript being drafted already, we actually use a statistical deconvolution method to deconvolute the PBMC to subtype specific and calculate the epigenetic age acceleration at a subtype level and sort of look at the temporal association, sort of prior exposure of different cancer treatments and resulting different epigenetic age acceleration and further link to the downstream chronic health conditions. So the subtype analysis really lead to the ongoing pilot study, a single-cell genomic accelerated aging. So we want to characterize the variability of cellular aging at a single-cell level and evaluate the modes of change whether the CBGs are asynchronous or synchronous across all these sites, the CBG sites that included in the epigenetic clock, and evaluated the transcriptional implication of these clock CBGs. And then the single cell discovery may yield novel primary target genes and the set of co-regulator genes for new anti-aging interventions. I want to just mention briefly these are the couple of papers that got my attention. Really, these are very elegant in my mind concept and study just published last year, recently in the Nature Aging, along with a commentary. I would recommend you read this if you are interested in this topic. And if you are an AI fan and interested in epigenetic clock, you may want to read this GPT-based aging clock. My group tried to explore this last year, and we continue to explore this and try to expand to see whether we can discover anything better than the popular ones. Well, several years ago, I submitted this grant proposal to examine and integrate these aging biomarkers, different aging biomarkers, to evaluate the influence of non-modifiable. These are, for example, genetics, race, ethnicity, and sex, and cancer treatment, and also modifiable as social environmental factors, health behaviors. They act on these aging biomarkers, and through that, leading to the increased risk of chronic health conditions. Although this was not funded after several years of trying, but nearly everything in this grant has been published except for the integration part. And this is a sort of serving as a contrast. Just received the funding decision from NCI, and the aim one is to look at the effect of health behavior and social determinants of health on the rate of change. So that we look at longitudinally, look at the rate of change, the trajectory of the epigenetic age acceleration, and to look at the social, environmental, and behavioral exposures. And also important to evaluate the association between the rate of change and the subsequent long-term outcomes in the vulnerable survivor population. And we hypothesize the rate of change, if it's a greater change, it's predictive of future onset or increasing severity of the specific chronic health conditions. And M3, I think this is sort of make this sort of, I think it's one of the most important success factor where I introduce into not only the observational study, but also two intervention studies that we introduced. One is exercise intervention, and the other is sleep intervention. So we will collect the samples prior to intervention, at the end of the intervention, and also a short duration of follow-up. Try to see whether this health-promoting intervention behavioral modification delivered through the clinical trials. Original design was improve physical fitness or sleep quality, but I want to evaluate whether the aging can be slowed down, if not reversal. So, what's next after this unprecedented progress of a decade? I think that the genomic era really provided enormous opportunities in clinical cancer survivorship research, and future perspectives should be emphasized in order to accomplish the goal of novel discovery, and may serve as a paradigm for genetic genomic research involving other populations at high risk for long-term outcomes. So first, I want to mention multidisciplinary collaboration should be fostered. This arrow pointing both ways. So on one hand, mechanistic study conducted by basic scientists using the mouse model, using in vitro iPSC system may inform the clinical research. And on the other hand, findings based on population science research can nominate candidate genes for further basic science follow-up investigation. So that's one. And the second is molecular-based profiling should really be conducted using the longitudinal follow-up. So along the cancer continuum for childhood cancer survivors, the primary cancer diagnosis lead to accelerated biological aging, and it remains to be assessed whether accelerated aging really contribute to the chronic health condition, which include the subsequent, we call it second cancer. So a lot of these survivors will develop as part of this chronic health condition, the subsequent adult onset, subsequent neoplasm. And notably, there's, if you know, there's the Onco-Aging Consortium, and they formed by the National Cancer Institute and the National Institute of Aging in the US. And I was also a task force member for the AACR. So we study really sort of looking into aging and cancer and aging. So cancer can accelerate aging, and accelerating can lead to development, initiation of cancer. So this is, the aging is the #1 risk factor for carcinogenesis. And thirdly, in this so-called applying the application of cutting-edge data science technology for in-depth exploration, like using machine learning and deep learning method, is super important, and future research may be accelerated by applying these methods into the rich omics data set. So with that, I will end with acknowledging our Cancer Center survivorship program at St. Jude, and department leaders, a lot of my collaborators, and my former and current trainees, as well as grant supports. Thank you for your attention, and I can start. We can use Chinese, if it's better for the question and answer. So I also want to just mention that I have multiple because I got more than around $12 million as a grant support. So it's a lot of budget to spend on a lot of research, different research directions, and the aging biomarker is one of them. And I also have 2 other directions. The other is genomics, genetics, precision medicine, and integrated genomic study on the cardiotoxicity, the treatment cardiotoxicity. And the third one is pediatric cancer genetic etiology study. So postdoc fellows or staff scientists, if you anyone or you know anyone interested, please recommend to me. And in fact, I want to thank the CGM because just today I am super excited encounter with a potential candidate for filling this position already. So I'm super happy. Thank you. Thanks.
非常感谢王老师刚才非常精彩的报告啊，我们也期待啊，今天有没有这个主动联系王老师的啊？听到王老师这边这么多的 funding 啊，我也是非常激动。然后大家有问题的话呢，可以直接开麦跟王老师进行一个中文的一个交流。
我就先讲了啊，因为这个 chat 里面有一些 question，就是说我可以说，就关于那个端粒长路，有人问，就是说端粒酶活性的问题，这个问题呢，就是说是早期的时候是有过这种就是说去intervene 这个，就是说对于端粒长度。但是端粒长度是比较要小心的是什么呢？端粒长度长是一个 oncogenic events， 所以就是说它是一个 double sided，就是双刃剑了。就是说怎么去更好的去控制这个是到目前为止没有很好的就是说确定性的方案，就是说你正常锻炼同时会增加癌症风险。所以这是一个比较 tricky 的地方。嗯，药物干预可以取得长寿。药物干预是可以取得长寿的，这个是很有趣的问题，因为我自己正在做。我可以提前透露一下，就是说，我们流行病系呢，主要是做这个行为方面的，所以行为关系最好的，你知道，就毒副作用，你不用怕，不用担心，对吧？像我们有 physical therapist，他是 Kiriness，他自己就是 physical therapist，所以他会做 exercise 的 intervention。那我们也做 sleep intervention，因为有 psychologist。然后也做这个 mindfulness，就是冥想啊。然后我也正在跟别人合作，准备设计 diet，就是饮食营养的 innovation。那说到药物干预呢，我们就是说，我现在正在做的有提前拿到一些结果的，就是用这个metformin。metformin 是叫二甲双胍还是什么，就不知道中文是究竟怎么说。它是一个type 2 diabetes 的药物。嗯，是降血糖的。呃，但是它有如果 FDA 它没有 approve 这个off-label use，就是说不能 officially treating，因为 aging 到现在为止 FDA 不认为它是个 disease，所以这就是没有一个药物，现在到目前为止没有这个药物是完全针对 aging 的。这个因为 aging 没有被 define 成 disease。所以就是说对每个人来讲，大家衰老是inevitable，对吧？我们每个人都是一定是会衰老的，但现在目前我觉得比较 realistic 就是怎么 slow down，就是 healthy aging，就 instead of 增加 lifespan，我们是想增加 health span，就是我们能够更加健康的活着。所以就是说，药物干预，我觉得 metformin 很有前景。不光是因为我自己在 cancer survivor 里面做，我为什么要做这个呢？因为 cancer survivor 我刚才讲了半天，就是说主要是突出它比我们要快速衰老。如果用 physical frailty，就是人的这个体弱来比的话，它是30年，这个是很大的一个 gap，对吧？那如果你用这个端粒长度，它是11年。你如果用 epigenetic age，它就是另外的。那你如果用 clonal hematopoiesis measure 这个 prevalence on average，它是20年。所以这个属于高危人群，所以这个更加是 justifiable。你可以去做这个 trial。那我们现在已经拿到了一些数据，那底下要准备 expand 做更多的研究。基础代谢率跟长寿是有一定的关联的。我不是这个上面的 expert，但是呢，就是说代谢intuitively 这是肯定是合理的。啊、端粒长度检测金标准还是 Southern blotting。我是因为用了 whole genome。其实 NIH 在5年之前、6年之前曾经有一个 grant，它就是要来，就是说人家 propose 怎么去测量端粒长度。所以这个是一个虽然是一个 old story，但是一直是一个 debatable 的一个东西。那就是我是用 whole genome 去用那个两个边端的那个 reads 去 count 那个 motif 的那个长度。那跟那个如果你是做这个 population scale 的，不是 individual level 的，就是我是指的是 relative speaking，对吧？相对来说的话，你是个相对值的话，那这个用 whole genome sequencing 是可以去 estimate，因为现在大部分 cohort 数据biobank 那些数据都是有whole genome sequencing 的 data。那 Southern blotting 还是一个金标准。我自己比较过Southern blotting 跟那个whole genome derived telomere length，它的 correlation 是0.7。epigenetic age 是如何定义的？epigenetic age 和 SNP 做 GWAS， 还是 epigenetic 位点和 SNP 做 GWAS？ 是 epigenetic age 和 SNP 做的 GWAS， 不是它的位点。你其实提到了一个很有意思的问题，就是说现在这个 epigenetic clock 它有 这个我刚才讲了2个 generation，对吧？其实现在已经进入第4个 generation，第3个 generation 我基本上 skip 了。主要是pace of aging，就是说它是不是 predict 某一个点，是 predict 变化，所以叫 pace of aging。它选择的 demethylation sites 是 predict 它的变化速度。那个 generation 呢，就是说做的人比较少，但是现在做的第4个 generation，现在做的除了是我前面讲到的，就是用这个 omics 一起去 model，不光是只看 epigenetic layer，你可以看 metabolites。那别人已经 published 的metabolic age，它跟 epigenetic age 的 correlation 是0。这0意味着什么？意味着就是说 aging，我们修了那个大转盘，对吧？它12个 slice，那么这个12个 slice 里面有一些之间是 inform each other，它就是 correlated，但是有一些是独立的，所以就是说像 metabolic age、epigenetic age 它是不同的。那这个其实是 composite，最终就是说我们需要把 multi-omics 放在一起，然后用比较 sophisticated 的 model，不是用简单的线性的模型去select 这些 predictive sites 但是这个里面我想就是 quickly comment，就是关于这个 methylation sites，我刚才就是说想说没说的，就是一直到现在为止，除了就是说 Nature Aging 那篇文章，还有就是说另外一篇文章我可能没有包括进来的，就是说他们有一个预筛选，就是本来我们做这个选择 epigenetic age informative CPG sites 的时候呢，我们是 agnostic 的，我不管它的 function。那现在呢，大家要建一个 function 的 clock，那怎么做呢？就是做这个是 causative 的，它就是把一些 causal 的 change，比如说用Mendelian randomization 就知道这个genetically 它是相关的，就是说然后去先把这个 CPG sites 扫一遍，然后generate 一个 candidate pool，然后用这个 candidate pool 里面再去建这个 predictive model。所以呢，做到最后就是说你的 methylation 新的这个 epigenetic clock 里面，它是 causal 的这种 methylation variation instead of 就是说 agnostically picked 啊、所以这个也是由于当初这个 epigenetic clock 建当初，因为 epigenetic clock 本身这个 concept 才10年，也不久，就是10年之前是 Steve Horvath 是 inventor，呃Levin Morgan Levin 是他的 postdoc，那么他们2个人现在在加州的一家公司做 longevity 方面的，那就是说除了这个 biomarker 去 measure 这个 biological age 之外，也有一种办法，就是说你去怎么去找到这个机制性的东西。所以在industry 有好多的 interest，就是因为 longevity 很重要，对吧？那我说 longevity 其实还是 healthy aging 可能比较科学一点，就是不是为了不是为了 lifespan，是为 health span 来考虑。不同的组织器官衰老速度是不一样的。我们现在研究的 epigenetic clock 用血样或者用多组织样本也有见的。那主要是考虑 systemic effect，那这个 tissue organ specific 的这种是很重要的。有人问过我这个问题，就是我觉得要就是比如说像 skin 有 skin 的这个呃你知道 cosmetic 现在这个大家都很注意嘛，就是你皮肤保养是吧？这个美容，有这个公司叫Elysium，你们可以查那个公司中文翻译叫极乐世界，Elysium，ELYSIUM，我可以在这个里面 type，everybody，是这个。我是一个 academic 的，在 academic，但是我也是比较关心，因为我是to be really honest，我跟那个Morgan Levin 有一些communication，因为我当初是fascinated by Steve Horvath's work，因为 Steve Horvath 本人是一个 mathematician，他与 biology 没有任何关系，但是他是提出了这个methylation clock 这个第一人。那么他们 Elysium 这个公司，它就是说就像它它可以寄个 kit 给你，你可以知道你的methylation age 啊、那么它可以检测这个 skin 的 age，然后怎么去保养，就是说这个就是一个 example，就是对这个前面谢谢大家，就是给的这个一些建议啊。UK 八 bank 有UK 八 bank 我记得是有的，但是不 sure。但是 TopMed 就是美国的这个 TopMed 队列发了很多文章关于端粒长度的文章，因为他们是 whole genome sequencing 的，他们用这个找到了很多genetic association，就是他们把这个用 WGS estimate 端粒长度之后，因为有 WGS derived genetic variation，做了这个telomere 的 GWAS，是一个很大的发现。嗯，然后哦，这个很好的问题，就是说这个 epigenetic clock，我不知道应该不应该透露，反正我觉得也无所谓。就是作为人群的差异是很重要的。就是 epigenetic clock so far 都是 build on 这个我不知道其他地方有没有。我们关心的中国人就是 East Asian，呃，这个很重要。就是说我知道有人 test 了这个 clock，把欧美人群记忆的 clock，用中国人的人群去 test，效果还是不差的，比我想象的要好，因为我原来想象可能是用不了，呃，所以但是我也帮助别人在做，拿到了课题经费，在建呃针对亚洲人群的 epigenetic clock，嗯，organ specific age，哦，这个是一样的。age-related disease AMD age-related macular degeneration，这一类的模型就是我不是很清楚哎，我也在就是这种有可能这个 macular age-related 不是所有的 age-related disease 与呃呃这个 epigenetic clock 是关系特别相关特别好的，嗯，所以有人研究那个 dementia，就是这种Alzheimer's disease 是有人研究的，它是与这个 neurocognitive 是有一定的关系的，这是嗯卖的后面的主要发展，我是想我在 Tom 我是 Tom 的 investigator，但是我是属于加进去，我是做癌症的人，那 Tom 的是 NHLBI 的 cohort，我用它主要是在用它用来做 control 用的，所以说属于这种附带用的，不是这个主流。呃，那作为我们做 genomic analysis 的，我跟他们有个合作，是做那个呃做这种 structural variation 做这个就是我们前面讲的那个 chip，就是 clonal hematopoiesis，对吧？clonal hematopoiesis 可以是单点的，也可以是 large structural 的 alteration 那个我是做的。我主要是就是说看那个，我觉得对于 NHLBI 它是搞 lung 搞这种 blood、搞 sleep 这些 disease，对吧？cardiovascular disease，这个他们应该还在做，呃，做的那个我是今年2月份嘛？对，今年2月份 TopMed Consortium Investigator Meeting 在 DC 开的，我觉得里面主要的是现在是用 AI，我印象最深的是斯坦福的一个教授讲了一个就是叫Foundation 这个 model，用了 single cell 建的，我觉得那个是最promising 的一个东西。 没有
王老师，
谢
还有一个问题，
谢你咨询。
您可能 miss 掉了，就是从寿
我
命的角度讲，就是儿童癌症的这个幸存者，他的平均寿命是一般会小于这个没有患癌症的人吗？喂，
对，是的，他们这个死亡是mortality ratio 是高多了所以他们的寿命就是说不光说是健康，就是整个的这个死亡也是早的。那我去年其实说到这个呢，我去年如果既然也是这个场合嘛，我去年是在《Lancet Oncology》发了一篇文章，呃去年10月份，那个文章我特别喜欢，因为为什么呢？我不知道参加会议的是就是说有多少做实验的人，还是多少是 dry lab，就是做这个data analysis 人。那篇 Lancet Oncology 文章，我是 data reanalysis，是 feel very proud，就是没有投入一分钱，除了是我们这个，当然我们智力投资跟那个时间投资是另外的，但这个数据都是已有的。我就分析了这个带有遗传性这个 cancer predisposition 的这些carrier，他们不光是得 second cancer 的肿瘤风险增加很多，而且他们最终会死于 second cancer，所以是那个呃就是那篇文章主要报道的是这样的一个结果，所以儿童肿瘤是10%都是由遗传性造成的，on average，当然不同的这个 subtype，它的这个遗传度还是不一样的，像那个眼癌的话就会很高，那白血病就会比较低一些，所以就是说，就 mortality 来讲，是的，他们的这个 lifespan 是比这个 general population non-oncology population 要小很多。对，
OK，就是您讲的那个，它死亡率，那大部分是因为就是同一种癌症复发，还是其他癌症，或者有其他的疾病吗？
哦，对，这个我澄清一下啊，因为我们那个文章标题是 late mortality，late mortality 的定义就是说它不是死于，它就是成人之后了，就是说就是说这些里面我们把死于儿童肿瘤复发的人都是去掉的，呃，就是都是 survive 5年之后进入我们队列，然后如果是死于 relapse，还有这种再 relapse 那些人也去掉的，所以他们死于肿瘤都是死于成人肿瘤。我是说的是cause specific 的 mortality，当然它还有其他死亡。那第一死亡原因就是说因为疾病死亡，第一死亡原因是 second cancer。
哦，OK，稍微等一下。我还有一个
嗯，
问题就是，就是关于这个表观年龄的，然后它主要是用于这个成人的比较多吧，一般建模型的时候，我不知道就是您用在这个儿童这里
哦，
面，就
哇，
是之前有用的
这你这个问题
多吗？它
真好。
的性能怎么样
对，
啊？
对你的问题很好，我正在做。嗯，就是说你说的很对，就是说 model 这个其实我们用了他们做的 model 去看这个小孩的变化是很快，对吧？就是说 children adolescent，我现在正在做的一
对，
个什么事呢？做的是就是说把 infant，从这个刚刚生下来几个月大，一直到 puberty 生长发育到12、14岁，在做我叫它 developmental clock，不叫 aging clock，不叫 aging，是就是 developmental，所以在这个 stage，究竟是你说我们是叫它 aging 还是叫它accelerate aging 还是叫 accelerate development，对吧？
对，嗯。
所以就是说你 apply 你说的对的，就是说那个已有的 clock 没有任何是针对这个年龄段的，他们的样本基本上都是大人为主的，
那就是您实际上用的这里面就评估过它的这个，比如说它的，因为它是回归模型嘛，MSE 之类的这个性能在
评估
儿童里面。
评估过，对我自己
嗯，
做过，我自己的 personal experience，就是说如果你用 chronological age 来作为endpoint 来 model 的话，它的
对，
这个准确度是很高的， 就是说这个 epigenetic 就是 methylation 对于chronological age 的这个预估的能力特别强，所以就是说
OK
基本上就是你不需要用什么复杂的算法去做就可以了，
啊
就是
，OK
对，嗯。
啊，那就实际上这个表观年龄它基本上不怎么考虑年龄这个范围，都能用，大体上来讲，可以这么说吗
现在我用在那个低年龄段是行的，但是我觉得可能那个也许是一个，就是说不是一个 optimal 的，它可能是 suboptimal，所以我是正在建那个发育中，不是这个衰老中。
？OK，好，谢谢。还有一个最后一个
发育中，
问题就是，好，您
对你
说，
说，我
呃
说发育中我就选的年龄是
，OK，
neonatal 到
嗯。
到12、14，嗯。
OK，好，谢谢。第三个问题就是您刚才就是基本上文章里面都用到了一个队列，您能简要介绍一下吗？不太了解这一块儿，就包括
哦，
它里
可以。
面有什么数据啊、开放度什么的。好，谢谢。
嗯，可以，没问题。St. Jude Life，这个队列是就是是 St. Jude Children's Research Hospital 建的。那就是我们来的方面主要是institution 和那个和那个嗯，和那个NIH、NCI 的 funding，就是说我们一直都是有NIH 和那个 institution 两个支持的。甚至 Life Overall 这个 study 现在目前我们有6000个人，eligible 的有1万，6000个人是 enrolled 在这个 study 里面。那已经产生的数据 whole genome sequencing，就是说有5000。就是我已经 publish 是4400多，在 JCO 那篇文章是 2020，那就是说已经这个几年里面我们又产生了500多，所以就是说5000个 whole genome sequencing，5000个 whole exome sequencing，那我在做这个 methylation profiling，也在做5000个人，那 RNA-seq，我的那个方面在产生，就是说差不多会3000左右，然后所有的 functional genomic data，所以 genomic data 我都会有 multi-time points，就是因为它是变化的，对吧？所以你这个 methylation 跟那个 RNA-seq 啊、它是会变化的。我们同时这个 cohort 也在做 proteomics and metabolomics，另外的 faculty 在 lead 这个。那我自己对 metabolomics 我是感兴趣的，我自己的独立的做metabolic age 这个pilot 啊、那这个其他的这个就是 molecular data 这么多了，对吧？那我其他方面呢，我们也在做一些就说是都是一些尝试性的，就说已经有 data，你可以 search 那个 s e n j u 的 cloud，如果你 Google 就是说 online search s e n j u 的 cloud，那 s e n j u 的
嗯
cloud 上面有一个叫 survivorship portal，那个 host 在那个上面，它有一个入门，有一个 landing page，你可以找到 survivorship portal，那上面你就可以access 这个access 这个s e n j u Life cohort。啊、它的好处就是什么呢？就是说它又有 clinical data，就是我们这个什么什么病啊、什么年龄啊、什么race ethnicity，这些 outcome，它都可以access。那同时也能access genomic data。我们 genomic data 有 genomic browser，有那个 variant browser，就是怎么说呢？就是说你想 play with it，还是 seriously analyze it，就是两种方式都可以。就是说 bulk，我说的就是 bulk download，或者是你想 play，就是想去，比如说你有一个发现想 look up，对吧？那
嗯
你可以在那个survivorship portal 上面可以看。我们data genomic visualization 做得很好。
，OK，就有涉及到费用还有可接触的人群吗？比如它有什么限制吗？
没有限制，我们只要就是说，你如果是access 的话，那就是说 browsable 的 data 是完全是 free 的。那如果你是想做 publish something，做一个 project 的话，你
嗯，
要写这个你的 contemporary use，就说你想做什么，对吧？就
嗯，
相当于
对
写一个一页纸的一个 proposal。
，proposal，嗯，
对，然后我是data access committee member，就是我们一般的 process 是，就是说你 submit 了之后呢，这个会给我们这个 committee去 review 就是我们没有一般是就是说 so far 没有任何原因去 reject application，然后 access 是 total free 的。
哦，OK，这太棒了，好，谢谢。
嗯，对，不客气。
好的，非常感谢，王老师今天非常精彩的报告，然后也给我们进行了一个非常详细的答疑，那我们后面的话也再赘述一句，有希望去王老师那边做 postdoc 的，可以直接跟王老师联系。那在王老师汇报当中提到的 Nature Aging 上的一篇文章，Causality Enriched Epigenetic Age and Couples Damage and Adaptation。那我们在6月1号呢，也邀请到了殷可君博士来给我们进行一个报告。那我们本期的报告就先到这里啊，非常感谢王老师，也谢谢参加的各位朋友。那我们
谢谢。
下期再会哈。
谢谢。
嗯，谢谢王老师。